Great. Hi, everyone. Thank you so much for coming to the last session of the 2022 Leading Edge Symposium featuring developmental biology. My name's Jen, and I will be one of your moderators today, and you'll hear from Sumru, the other moderator, a little later. So a few scheduling details to go over before we begin. Unfortunately, Elle Robertson, who was our third speaker, will not be able to give her talk in person today. However, a recording of her talk will be available later on the Leading Edge YouTube channel. So please check out her exciting work on the role of hedgehog signaling in the regulation of uterine smooth muscle remodeling during the estrus cycle a little later. And a few house rules before we begin. Please mute your microphones during the talks so you don't interfere with the speakers. And if you want to ask the speaker a question, there are two ways in which you can do so. First, you can either type in your question during the talk in the chat, and we can ask this after the talk is over, or if you would rather, please raise your hand after the talk and either the speaker or moderator will call on you. Great. So now I'm really excited to jump into these last final set of talks and introduce our first speaker, Val Tornini. Dr. Tornini was a NSF graduate fellow in the POS lab at Duke University in the Department of Cell Biology, where she studied the cellular and molecular mechanisms of tissue regeneration. Now, as a postdoc and Hartwell Foundation recipient in the Giralda's lab at Yale, Dr. Tornini studies the code of specification in vertebrate central nervous system development. So I'm really excited for Val's talk. We'll get that started.
Hi, thank you very much for that introduction. My name is Valerie Tornini. I'm a postdoctoral researcher at Yale School of Medicine, and I am excited to tell you today about big roles for tiny peptides in vertebrate neurodevelopment. And so we start with how I think about cells and cell identity and how much of the cell states are determined by
Jen, on mute.
RNAs, around 80%, and a very small fraction is these coding RNAs, protein coding RNAs. And one thing that when we look at one of these aspects, which is protein size, here we're showing just on the x-axis, and we look at the abundance, we notice a really strong drop-off between 100 amino acids and below. So we're missing a big chunk of these proteins. And the question is, well, is this real or is this just an artifact? And it turns out that although there are less than 4% annotated proteins that are 100 amino acids or smaller, around 10% of the proteome is posited to actually encode for these small peptides. And so when I started, we said, okay, well, what if we expand our where we're looking for some of these proteins into the non-coding RNAs transcripts? And this kind of started when we looked at a lot of the long intergenic non-coding RNAs that were identified in zebrafish, which is my system, my model system for vertebrate development. And through ribosome profiling and other techniques, there were actually these encoded, these micropeptides or small peptides encoded within a lot of these long non-coding RNAs. And especially they were enriched in these developing cell or tissue specific manners, and especially in the central nervous system or brains. And so this is where my project started. I wanted to ask, what are the roles of micropeptides in vertebrate development? And my talk today will go through a pipeline of that I've developed, looking at essentially screening for effects on development and specifically neurodevelopment using behavior as a readout, then looking at their effects on circuits and pathways, how cell states are affected, and finally, the molecular mechanisms that are causing any of these phenotypes. And so to start, here I'm showing how we screen for behavioral phenotypes of micropeptides using zebrafish, just F0 knockouts. Here we have a video of single larvae between 4 and 7 days post-fertilization in the different F0 candidates. And zebrafish in this system is really can exquisitely define and capture a lot of the quantitative aspects of zebrafish behavior and locomotion. So this is a really great way to screen for phenotypes. One of the ways that we do this is looking at these different parameters and how much they deviate from their respective controls. And two of these genes that we looked at, LINK-MyPep and LINK-WRB, had actually really similar profiles, which is a bit striking. And here we're showing just this hyperactivity during daytime. And the other interesting aspect of these was that both of these together had this ultra-conserved sequence in their 3' end of the transcripts, suggesting that they actually might be related. So then to go on and actually definitively show that these effects are caused by coding versus non-coding effects, we had to generate stable mutants, here defined by these different cartoons, to actually say these are caused by the coding, the non-coding function, etc. So long story short, looking at stable mutants for these, here I'm just showing the final product. So LINK-MyPep and LINK-WRB protein coding, but not non-coding, double mutants are hyperactive. And here I'm showing on the bottom, it's just single traces for 1 fish per well at 6 days post-fertilization compared to their wild type. So this is really, and it's really quite extreme hyperactivity. So then we asked, well, why are they hyperactive? One way that we do this is to identify circuits and pathways that are altered in mutants using something called behavioral fingerprinting. So we take those parameters that I just mentioned before, and we normalize all of those to wild type levels. And then we ask how those levels are affected in the mutant, either up or down from the wild type. And when we do this, we generate this, this fingerprint. And the question is, how do we get to the mutant fingerprint? Well, so Jason Reel in his postdoc and my collaborator did this screening through over 500 FDA approved small molecules on wild type larvae to generate those same fingerprints. And we said, which of these match most to the mutants? So this is what that looks like, those 500 FDA approved drugs and the fingerprints for these. And the LINK-MyPep mutant fit into this category over here. And when we look at the correlating drugs and test them in vivo, we ultimately find that the mutants that we generated mimic NMDA receptor antagonism and are really sensitized to glucocorticoid receptor activation. So at this point, we had to say, okay, why are these circuits and pathways being affected? And we had to know a little bit more about what the gene may be doing. So what do these genes encode? LINK-MyPep and LINK-WRB have homology to high mobility group N1 or HMGN1. And here I'm showing the amino acid sequence of the human flanked by either of the 2 zebrafish transcripts. HMGN1 binds directly to nucleosomes with preferential binding at DNase1 hypersensitive sites. And using that really interesting 3' UTR and other genomics techniques, we actually identify unannotated related genes in vertebrate species only across 550 million years. Importantly, for neurodevelopmental disorders and research, this gene is overexpressed in Down syndrome or trisomy 21 and has been identified as an autism spectrum disorders risk gene. So now understanding that this may be involved in chromatin and gene regulation, we ask, what are the effects on cell states? To do this, we next perform single cell RNA and ATAC-seq to reveal effects on cell states. And here's just a simple cartoon looking at the experimental setup. And when we plot each of these cells on a UMAP, we actually see a quite strong overlap and really, really minute differences. So we teamed up with the Krishnaswamy lab to identify the most affected cell states using some of their algorithms called MELD. When we do this, we find that cerebellar cells and cerebellar granule cells and oligodendrocyte progenitor cells are the most susceptible to loss of these double genes. And here we're looking at the most statistically different attack peaks or chromatin accessibility peaks, and here defined in yellow as being more accessible in either wild type cells or link-MyPep cells per cluster to these clusters. And when we cross these double mutants into these OLIG2 GFP transgenic fish, which would especially define these oligodendrocyte progenitor cells and other lineages, we find that especially in the cerebellum, here pointed by the arrow, we see a really significant depletion of these cells. Why was this really interesting to us? Well, both of these, both the cerebellum and myelinating oligodendrocytes are posited to be these jawed vertebrate innovations. And here I'm showing just a cartoon of the lamprey brain, which is a jawless vertebrate, and the zebrafish brain, the emergence of the cerebellum. And so one of the cool experiments that are underway and results to be determined, we teamed up with the Marduk lab to introduce into lamprey either link-MyPep or human HMGN1, and we'll perform ChIP-seq and ATAC-seq to assess for transcription factor sites that may be now becoming accessible for these OPCs or cerebellar cells. So to summarize, we found that through screening a lot of these micropeptides, we found that link-MyPep and link-WRB loss of function mutants are hyperactive, that these mutants mimic NMDA receptor inhibition and are sensitized to glucocorticoids. And although we have a globally altered chromatin accessibility, we find that the most susceptible cell types to these are evolutionarily newer vertebrate cerebellar and oligodendrocyte cell types. And I'm happy to share that this is now out on bioRxiv, so I look forward to community feedback from that preprint. So as I move forward in my postdoc and into my own lab, I look forward to identifying other cryptic micropeptides and functions in neurodevelopment, as a lot of these may actually be performing some really cool functions that maybe larger proteins are not quite able to do. But also thinking about chromatin regulation, and especially the results from these genes, we're reminded that a lot of chromatin modifiers and remodelers are also implicated in neurodevelopmental disorders, and especially in my case, autism spectrum disorders. And so I've generated mutants for these and other candidates and have done the similar pipeline to try to understand across the scales, looking at cell and subcellular states, cell identity, circuits and pathways, and ultimately effects on whole organism and behavior, the effects of these micropeptides and chromatin modifiers and remodelers on cell states. And with that, I like to pause here and acknowledge my advisor, Antonia Heraldez, my collaborator, Jason Reel, all the other authors, the Yale Center for Genome Analysis, and my funding sources, and you for your attention. I'm happy to take any questions.
Great. Thank you so much for that talk. Let's see. You already have some questions in the chat, Val. So we had a question from Helen Wilsey, which says, are there small peptides expressed in a cell type specific manner during brain development?
Hi, thank you for that question. So this is quite ubiquitously expressed, and in the preprint we show by the clusters, you can actually check the expression. They are pretty broadly expressed, a little bit more in some neuroprogenitor cells, and it seems to be more enriched in Purkinje cells, but across the scale, all cells and in general, all vertebrates seem to have expression to some level in their nucleus.
Very cool. And then I had a really naive question. So what is your definition of a micropeptide? Like, how do you actually know that it is by itself like a protein?
Yeah, so that's excellent. So it's pretty Vague and broad. It's so far has been that it's less than 100 amino acids, and that's partly due to just computational resources and just tools that have been able to identify them, definitively identify them. In our case, we had ribosome footprinting data, and we also generated custom antibodies to show that they are generating proteins and that they're abrogated when we have the loss of function mutants.
Yeah,
but and now it is rising. A lot of people are really looking into finding them, defining them, and then actually finding their functions. So it's a really exciting time for that.
And then Sumru has a question.
Yeah, thank you. Beautiful talk, Val. I was curious, this cell state analysis you guys done with single cell RNA attack seems very powerful. The two cell types you pick up is one is an excitatory neuron progenitor, one's an oligodendrocyte progenitor. I was curious, was the genes that are the program you observe in your mutants overlapping? To what extent it was overlapping in these different two cell types, or it has completely different gene sets that is affecting?
Yeah, generally speaking, this is something that we're still analyzing. Generally speaking, they're not derived from the same progenitor pools or anything, and they do have distinct gene regulatory networks that establish them, but there are some overlap, so that's something that we're exploring. The other aspect is that they like physically are in the cerebellum, like they are interacting, and so one of the things that we're also exploring is whether it may be that the function, the dysregulation of function of one cell is also maybe affecting the other. So it's something that we're exploring just with other experiments, including HCR type experiments.
Cool, very cool. Thank you.
I don't see any questions in the chat, so I have more questions. So I was actually really excited about this assay system of yours with regards to looking at like phenotypes in your fish. And so you talk about hyperactivity, but are there any other particular phenotypes that you're really kind of excited about exploring?
Yeah, so I didn't have time to show here. In the program we show the profiles for all the ones that we screened, and they can vary. So the types of parameters that we're looking at are locomotor activity, sleep, during daytime, nighttime, how long they sleep, how long it takes to wake up, etc. So these are very baseline behavior profiles. In the other work, we also look at things like seizure susceptibility, etc. So in terms of the micropeptides, there are different phenotypes that we see. And we just followed up on this one because it was really quite extreme, but some of the others also seem extreme and specific. So some of the others, maybe at some other point, maybe in my own lab, will find what those are doing. But there are other phenotypes that came out from this screen.
It's a goldmine. That's amazing. All right. So with that, I think we'll move on to our next speaker. So our next speaker is Natasha. Oops, let me just minimize my screen here. Natalia Shylo. So Dr. Shylo was a graduate student in the Weatherby lab at Yale University within the molecular cell biology, genetics, and development track of the biological and biomedical sciences program. Here at Yale, she studied the ciliary gene TMEM107 and how mutations result in ciliopathies as well as left-right patterning defects. Dr. Shylo is now a postdoc in the Trainer Lab at the Stowers Institute for Medical Research and continues to pursue her interest in understanding the mechanisms that underlie left-right patterning decisions. So with that, please take it away.
Thank you so much. Thank you, Jen, for such a kind introduction. So as she said, I'm a postdoc in Trainer Lab at Stowers Institute for Medical Research right now. And today I'm going to tell you about veiled chameleons and how they establish left-right patterning. So why do I care about left-right patterning? Aren't we fairly symmetrical? Well, it turns out that we're fairly asymmetrical on the inside. We have different number of lobes on our lungs on two different sides. And it's very important that the heart is on the left side of our body. There are some people who have all organs reversed. It's a complete mirror image. And a lot of times, as long as the organs have the proper positioning in relation to each other, those people don't even know they have this condition and they live perfectly normal lives. And you might even know some of those people, like Enrique Iglesias and Catherine O'Hara. The problems arise somewhere in between, when at any point organs hesitate which side is left and which side is right. So oftentimes the heart will get stuck in the center, and these conditions are known as heterotaxy, and they can be associated with extremely high mortality rate in the first year of life. So it's important for us to understand how left-right patterning is established. So the organ that is as important for establishing left-right asymmetry is known as the left-right organizer, or the node. In humans, it's this dot right here. Here it is in the embryo, right anterior to the primitive streak. In the mouse, the organizer has a teardrop shape. Here it is highlighted in an in situ. In the chicken, in this in situ, it has this kind of comma-like shape. And amongst these animals, there are two modes of establishing left-right patterning. Mice and humans, in their left-right organizers, they have motile cilia that generate left-right flow, and the result of that, we have asymmetric expression of genes on the left and the right side of the organizer. In chickens, instead, they use asymmetrical cellular movements to accomplish the same task. So it turns out that having cilia in your left-right organizer, motile cilia in the left-right organizer, is the ancestral state. Mice and humans have cilia in their left-right organizers, motile cilia, as well as frogs, zebrafish, and sea urchins. I just told you that chickens instead use cellular rearrangements. And so for a really long time, we had no idea what happens in lizards and snakes, and whether this loss of motile cilia was something that just happened in avians, or was it a synapomorphia of all reptiles. And it turns out we actually have a lot of unknowns as far as reptile development. In addition to not knowing anything about the cilia in the left-right organizer, we know very little about reptile gastrulation, neural crest cells, and really most early development. Why is that? Well, it turns out at the time that when lizards and snakes, the squamates, lay their eggs, the embryo is actually very well into their development. They've already formed the head, the heart, the somites, they already have limbs. So it's far too late to study gastrulation and left-right patterning. So that's why we're excited about veiled chameleons, because at the moment they lay their eggs, the embryo is actually at pre-gastrulation stages of development, which means we finally now have a reptile research organism to study all of these early processes. And we think they're going to make a great research organism. In addition to having those the embryos at early stages at the time the eggs are laid, they're also a popular pet. So there's established husbandry. They will lay eggs year round, and they lay pretty large clutches of eggs. I've been working on developing a number of techniques and resources for these. I started by developing an embryonic staging series. We now have several transcriptomes available for these guys. I can do cell culture and embryo culture, and obviously I can do live imaging with those. We've done a lineage tracing, and we've recently sequenced and annotated the genome. And I want to highlight that a little bit more. We're working on the website that will soon go public, as well as the associated publication with that. Now, the reason I want all those resources is to study left-right patterning. And over here, I have a schematic of this generic organism and how at the molecular level left-right patterning is happening. So what we're looking at here is mostly the left side of an organism. So in the middle, we will have the left-right organizer. Here it's named the node. Typically, it will be ciliated. There will be motile cilia. And somehow there is this initial signal that from the left-right organizer, it induces the expression of nodal on the left side of the embryo. And nodal is kind of the key molecule in this cascade. So that's why oftentimes it's referred as the nodal left-right patterning pathway. So nodal will auto-activate its own expression. It would also induce expression of genes known as lefty. So here lefty will be expressed in the lateral plate mesoderm, and lefty will be expressed in the midline. And lefties act as repressors of nodal. So they control to make sure that it doesn't get overactivated on the left side, but also doesn't cross the midline and induce its own expression on the right side. Furthermore, nodal will induce downstream factors like PIDX2, which is a transcription factor. And PIDX2, in its turn, it signals to other organs to say, okay, hello, you're on the left side, please make left side organs. So that's kind of a general overview of what this pathway would look like in a generic animal. So I wanted to figure out what it looks like in veiled chameleons first. And so very briefly, in chameleons, we actually have two nodals. They're both expressed on the left side, suggesting they're both involved in left-right patterning. Instead of just lefty acting as a repressor of nodal, we have a lefty that is expressed in the midline. So it only retained its midline barrier function. And Cerberus-1 is a molecule that represses nodal in the left side of the letter-like mesoderm. Furthermore, we have the PIDX2 that is expressed in the left side of the embryo, and then all the associated downstream signaling. Now, this is kind of a very quick overview of this, this molecular cascade, what happens here, but I didn't tell you, well, how is left-right signaling, how is this originally triggered? What is the original determination of left-right asymmetry? So, I told you that there's two main ways to do it. So in mice, there are motile cilia that generate flow, and in chickens, there are asymmetrical cellular movements. And since having motile cilia is the ancestral way, we decided to check first, well, are there motile cilia in veiled chameleons left-right organizer? So the easiest way to do that was to look at associated stages in development. So these are roughly the stages where left-right patterning is happening. And I looked at two genes that are required to make motile cilia, FOXJ1 and DNAH11. And I see absolutely no expression of either of those genes at any of these stages, suggesting that at no point in these early development stages are there motile cilia present. Therefore, there are no motile cilia in the left-right organizer of these animals. So if there are no motile cilia in their organizer, does that mean they're more like chicken? Does that mean there are these rotational asymmetrical cellular movements in the chicken, in these embryos? And how would we even look for them? Well, another thing that happens in the chicken is that the node becomes kind of tilted in asymmetrically positioned, so we can look for that. But also some of the molecules kind of become asymmetrically expressed as a result of the cellular movement. Sunny hedgehog is originally symmetrically expressed, but then effectively gets dragged over to the left side as the cells migrate over there. So I had a lot of techniques to kind of address this question. So we decided to start by actually looking for these cellular movements. So I developed a way to culture these embryos and take live imaging. So these develop very slowly. So this video actually takes place over a number of days. And we can see the blastopore here. We see the midline forming, the head folds, the foregut pocket. But in all fairness, in this particular video, it's really hard to see the moment where left-right asymmetry kind of when it gets triggered and when it gets established. So what we're going to do instead is we're going to look at this embryo so this is the ventral view of the embryo, and we're going to look as optical projections. So we're going to be looking at transverse sections effectively between these two yellow lines, and they're in the posterior end of the embryo. And so I'm going to play this video. What you're going to see is that this left side, as left-right patterning is there's a trigger, this left side just lowers down. And then after that, the right side then evens out. And then after that, the embryo looks perfectly symmetrical again, as far as the morphology of the embryo, just kind of in its appearance. And so at some point, there was this trigger and there's this morphological change that happened. Now we can look at the video and sort of see where, how the embryo looks like at that point and things like that, but we don't really know what's happening molecularly. So we wanted to sort of see whether we can match up what we're seeing in this video to molecular marker expression. So Sunny hedgehog was a great place to start. And so at the end of the video where the embryo looks pretty symmetrical, the left and right sides look pretty symmetrical, that's when we see Sunny hedgehog expressed fairly symmetrically in the embryo. And it's really cool to see that at stage 10 and 11, it is not expressed symmetrically. There is some sort of asymmetry that is revealing. It's very visible here in stage 11. I'm going to highlight it right here. So right next to the blastopore, there is this asymmetrical positioning of Sunny hedgehog. What's very interesting, and I want to really kind of emphasize this point, is that Sonic hedgehog is not expressed asymmetrically in these embryos. In fact, when we section these embryos and we looked at their notochords in their neural tube, in the floor plate, Sonic hedgehog is expressed equally in the left and the right sides of the neural tube. So Sonic hedgehog is expressed asymmetrically. However, there are morphological changes to this neural tube that we saw in the video and in the embryo itself. And in these sections, the way it presents is this tilt of the neural tube over to the left side. So this hinge point is tilted to the left side. And that's what Sonic hedgehog's staining is revealing to us. It's really the morphological change in an embryo. Now, how does it relate to all the other left-right patterning signals? It's really cool because I told you nodal is expressed asymmetrically on the left side of these later stages, but at that point, the embryo looks, as far as Sonic hedgehog morphologically, apparently looks fairly symmetrical. And it's really cool because we first see these morphological asymmetries when nodal is not expressed yet, which means that there is this morphological asymmetry that is somehow triggered in the embryo. And these morphological changes is what then trigger the asymmetric expression of nodal on the left side and the subsequent nodal cascade. So in summary, if you need some key points to walk away from this presentation, I hope you remember just 2 key points. Motile cilia are not involved in left-right patterning in veiled chameleons. And instead, asymmetry is established through changes in tissue morphology, and that change in tissue morphology then triggers molecular changes in the embryo. So there's lots of questions to address from now on. So in the future, I would love to figure out what are the mechanisms of left-right patterning in veiled chameleons and what is triggering these morphological changes. We now have the sequenced and annotated genome, so it's really time to start doing some genome editing and targeting some of the genes in this pathway. Furthermore, my really kind of long-term dream is to use these guys and establish a bedside program and use them as a model of left-right patterning and heart disease. And why do I think that's possible? So if we look at a mouse embryo, and I showed you the mouse node, it's right here. And I told you that the mouse node has motile cilia that generate flow. What I didn't emphasize is that in mice and most other animals, cilia are actually present on virtually all cells in their body. They're non-motile primary cilia. They're present in the lateral plate mesoderm, the midline, the somites. They're all over the entire embryo. However, when we knock out the motility of the node cilia, the effect is so strong that it's basically epistatic to any other left-right patterning effects we might be seeing from knocking out other cilia. So it's virtually impossible to study the roles for cilia in other parts of a mouse embryo and their roles in left-right patterning, which makes me really excited that I now have a research organism that does not have motile cilia in their left-right organizer, and instead I can address the functions of cilia in the left-right, in the lateral plate mesoderm, in the somites, in the midline, and also in the heart. So in the end, I would love to thank all the people who made this work possible. And if you have any questions, I think we should have a little bit of time left.
Fantastic talk. Yet again, we have another great question from Helen in the chat. She says, awesome. Can you educate us on the genome? Is it diploid and how large is it?
Yes, so the genome is diploid. It has, it's 1.8 gigabases. So that will be like 1N is 1.8 gigabases. We have figured out that they have XY sex determination, and it's not temperature dependent. And we've even narrowed down on the sex chromosomes. Yeah.
And then I saw in your leading edge booklet that I was reading that you've been working on some CRISPR in these animals as well.
Yes, so sort of baby steps for now. You know, there's so many things to do with these guys that, you know, I just need to get to that experiment. But there's a wonderful paper that shows how to do CRISPR in anole lizards, and everything in place. Just the time and me being there to perform that same surgery and kind of experiment in veiled chameleons. I'm like fairly certain it's going to work.
That's really, really exciting. Yeah, yeah, yeah. And then I know that this was one of your major questions at the end, but do you have any hint maybe as to what could be upstream of nodal that's causing these like mechanical differences in left to right? Because I think that's really fascinating.
I know, it's very interesting. And what's really different about these guys from the other organisms we have, because one could argue, well, chickens also don't have cilia in their organizer. But what chickens and mice have in common is they have the primitive streak. These guys don't even have a primitive streak. So it's completely different. So what we think as far as what's going on in chicken, the model and idea sort of brewing in the field right now is that perhaps there's something about, literally cellular chirality and like chirality of like actin filaments that make cells kind of move towards a certain size, and then as a group all of that like starts flowing in a certain direction. But how that might potentially translate into this kind of wobble movement that I have no idea yet.
Yeah. Val also had really great questions in the chat. She had the same question as me. What do you think establishes the asymmetry? And then she suggests things like maybe looking at the actin network or cell adhesion networks. Have you looked at that?
Yeah. I would love to do that. And we know that some of that has been shown to be important for parts of the pathway and signaling, things like that, but perhaps also, the origin of triggering this. And another question is, when something loses cilia, like when some mechanism gets lost, usually animals revert to a more ancestral form of mechanism. So presumably these guys have reverted to more ancestral form of establishing left-right asymmetry. So does that mean some of that is still present in mice and humans, and we just don't know because we're so fixated on motile cilia? It's quite exciting.
Yeah. And then there was a second part to Val's question, and that is, in your in vitro model, can you physically manipulate development, like pulling with beads in the opposite direction to see if that switches the direction of the...
Yeah, I think I absolutely have to do this experiment. It keeps suggesting every talk. We haven't done it yet. Right now, the way I image them is a little bit restrictive for me to kind of go in and manipulate them. A couple ideas of how I might be able to, and I think like adding beads will be not unreasonable. I think we can do that.
And then Terry Yamaguchi asks, how do the germ layers form in the absence of a primitive streak?
So it's a little bit. So the way gastrulation happens in these guys, the best we know is a little bit in between what happens in like a frog and a chicken, for example. So there is, so in frog, you would have the gastrulation occurring through the blastopore. So we have some of that, but also there is like not quite a primitive streak sort of motion happening there too. So there's a little bit of involution and ingression that's happening in these embryos. So it's kind of in between where, some of the more basic like frogs and zebrafish and things that have a primitive streak. So they obviously still gastrulate. But in between.
Very interesting. Really unique. It's just fascinating how different organisms have different strategies to accomplish.
I know. And it's
more or less the same
so
thing.
much by not looking at them.
And then Prina asks, could asymmetric RNA transport or degradation play a role in the asymmetric distribution of these proteins?
Oh, entirely possible. Yes. So RNA degradation is actually really important for left-right asymmetry in mice. So I'm sure there are processes that are happening here as well. I just don't know. I don't even know where to look yet, but we'll get at it.
Yeah. So many really exciting avenues to pursue. So yeah, that's been really great. We're going to take a quick 10-minute break right now. As I mentioned in the beginning, if you missed the message, Elle was not able to give her talk today. And so we're just going to take a little 10-minute break, and then we'll restart at 12:50 with Connie's talk. So thank you.
I think like Cara is more or less legal.
All right, welcome back, everybody. We're gonna continue with the second two speakers of the today's session. And first we have Connie Phong. Dr. Phong has, she did her PhD at University of Chicago with Michael Rust. And currently she's a postdoctoral fellow at Stanford University School of Medicine in the Farrell Lab. And she has a long-standing interest in understanding how organisms cope with environmental fluctuations and what are the molecular mechanisms of these biological adaptations to extreme conditions. So take it away, Connie. We're looking forward to your talk.
All right, thanks, Amar. I'm going to share my screen. All right, can everyone see my slides? All right, great, thank you. I'm very proud to be part of this class of Leading Edge Fellows and really appreciate this unique forum to sort of share the work that I've been working on and sort of get feedback on just the general research arc that I'm gaining towards or working towards as I'm trying to move more quickly into establishing my own independent research program. So I'm a systems biologist. I initially trained in computational and mathematical sciences before sort of making the leap into also doing experimental work in the lab of Mike Rust. I've always been interested in how the mechanisms by which organisms, biological systems interact with their environment must both be simple and complex at the same time. I think at the end of it, one sort of generous explanation of my life will be that I've tried really hard to channel that confusion into at least well-posed questions. So with that said, my sort of past, current, and future research goals are centered around three main questions. First, I want to understand how organisms collect, sort, and encode information about the environment, and that encoding being both at sort of biochemical and genetic level. Secondly, I want to understand how that environmental information then constrains regulatory systems for maintenance, growth and development, and reproduction. And finally, I want to understand how those regulatory systems will change over ecological and evolutionary timescales. So the work that I'm going to tell you about now in my postdoctoral phase of training is really in this category of problems. I'm interested in how embryonic development specifically contends with temperature. And for those of us who are trained more molecularly, I think it's easy to forget that organisms develop under sort of harsh conditions and the environment is both complex and dynamic, and that the mechanisms that guide maintenance, growth and development and reproduction are constrained and must function with respect to these harsh environments. Okay, So I think while most of us are sort of familiar with the fact that there are large seasonal fluctuations in temperature, there are also smaller, shorter timescale fluctuations in temperature as well. So what I've replotted here is data that's been collected at a weather station, the Harvard Force. It's not a very special week, but what it is meant to do is to highlight that within a given day, you can see anywhere of a 10 to 20 degree change in temperature. So these sort of temperature fluxes, whether they're happening on large seasonal timescales or short daily timescales, which are also called diurnal timescales, impinge on all aspects of normal organismal physiology. So things like circadian rhythms, something I worked on as a graduate student, also metabolism, intra and extracellular pH, and also developmental rate. So if you are a cold-blooded organism in particular, this means that you lack an endogenous thermal regulatory system. Cold-blooded organisms are also called poikilothermic organisms. So to contend with this 10 to 20 degree flux in temperature imposes some very challenging demands because you lack this sort of internal thermal regulatory system. And I think that this sort of the consequences of not having an internal thermal regulatory system are probably nowhere more profound than in early development. That's because animal embryos during early embryonic development go through a phase called cleavage divisions, where we start with a single cell that's been newly fertilized, and we're progressively cleaving the cell into multiple cells while conserving volume. But during cleavage phase, which lasts anywhere from hours in frogs to days in humans, the embryo is transcriptionally quiescent. So this means that it's unable to mount an adaptive transcriptional response to contend with environmental insults like temperature. The temperatures that I'm talking about also are sort of characteristic of the natural environment. They aren't ones that are in the extremal ranges that are known to trigger responses like the heat shock response in adult organisms. Moreover, heat shock response usually requires first going through the maternal zygotic transition. So really, these hours or days that an animal embryo is in cleavage division phase are pretty fraught. It's potentially dangerous. And without sort of the normal ways we can think of for an animal to sort of monitor and contend with this wide temperature range, what really has to happen then is that it's the innate molecular properties of the maternal contribution of stuff in the embryo that allows it to sort of progress successively through this dangerous part in early development. So I want to unpack that statement and get into what I think it means. But before I do that, I want to show you a high resolution data set of cleavage divisions and sort of my model organism of choice now, the temperate African clawed frog, Xenopus laevis. So these are data from the 2nd to 5th cleavage divisions of Xenopus laevis, and they were collected by Leonard Gellens, who previously trained with Jim in the Feral Lab and now has his own independent group in Leuven and is my collaborator in this work. So I've marked in this gnarly shade of green what is usually considered to be the optimal rearing temperatures for Xenopus laevis in captivity. And I've just plotted each of these circles. Open circles is a different embryo going through a cleavage division phase. So it's just the 2nd to 5th cleavage divisions collapsed. And what I want to extract from this data set is a developmental rate. So what I'm showing you here is the Arrhenius equation, which you may recall from sort of your earlier general chemistry courses. But what I want you to remember about the Arrhenius equation is that it comes from the theory of single biomolecular reactions, whereas development is a complex reaction, right? It's a superposition of many biomolecular reactions, both biochemical and biomechanical, and that are actuated by a heterogeneous array of macromolecules that are structurally and functionally different. But I want to talk about the developmental rate because I want to talk to you about the Q10. So the Q10 is a familiar quantity to circadian biologists, but I want to remind you that it basically is a quantity that describes how the rate of a reaction will change for every 10-degree change in temperature. So as you can see from the equations, the Q10 and the activation energies are related quantities, and sometimes I will use them interchangeably. All right, so this is what fitting looks like to get the Q10 for the cleavage divisions. Right, so I've been able to fit a straight line through most of the data points, including the data points that lie outside of this green range that determines the optimal rearing temperatures. So this is, if you step away from the immediate data, although I should say that the Q10 that I fitted here is of 3.12, which basically means that for every 10-degree change in temperature, the rate of cleavage divisions will increase by a factor of about 3.1. So what I meant to say was that if you step away from the immediate data, what you see, what is strikingly obvious is that you can fit a straight line through a lot of the data, right? This is not at all impressive, but it is striking. But I would argue that it is impressive because what it means is that all of the reactions that make up cleavage division, all of these biochemical and biomechanical processes, the superposition of all of these bimolecular reactions, they all collapse to a single temperature dependence, right? So a single coherent rate, like you would expect from simple bimolecular reactions, the reactions that are meant to be described by the Arrhenius equation, not something complex like cleavage divisions. So that is a phenomenon that I think is actually like non-trivial and deserves explanation, deserves a mechanistic understanding. But if you're not yet convinced by the fact that temperature scaling is a cool phenomenon, I want you to consider what temperature scaling looks like in homeotherms, or warm-blooded organisms like ourselves during early embryonic development. So it's known that elevated maternal temperatures of as little as 1 to 2 degrees during pregnancy can lead to spontaneous abortions and serious congenital defects. So what I've replotted here is one of the few data sets that I've been able to find for cleavage divisions in a warm-blooded organism. So sparser data points reflect the fact that cleavage divisions really do happen over a narrow range in warm-blooded organisms. But again, you see this sort of collapse to a single fit, so a single coherent temperature dependence. So a phenomenon that is conserved. So a future direction that I'm pursuing is really trying to understand over that evolutionary timescale that I was referring to, why the upper and lower limits of early embryonic development have changed from the transition from cold-blooded organisms to warm-blooded organisms, even though the sort of the core regulatory machinery is the same. All right, so the last thing I want to say in terms of trying to convince you that temperature scaling is a cool phenomenon is that there seems to be evidence that there's a universal Q10 for these embryonic cleavage divisions. So I've replotted here the data that I showed you before for Xenopus laevis, alongside data for its tropical cousin Tropicalis, also for zebrafish, and data coming out of Tony Hyman's lab in different species of nematodes. So what, again, is very clear is that these lines are parallel. They're straight lines that are parallel, meaning that they have the same slopes, meaning that they have nearly identical Q10s. So while I'm leveraging sort of the benefits of Xenopus laevis as a strong model organism to understand the underlying cell cycle regulatory machinery, whatever sort of biophysical mechanism that I find in Xenopus laevis will probably also be true in zebrafish, will probably also be true in these nematodes, because these are homologous processes and there seems to be evidence for a universal Q10. Another way of thinking about this is that I've taken the cell cycle regulatory machinery and used it sort of as a model system for how all non-circadian circuits in cold-blooded organisms must contend with this wide operational temperature range, trying to understand underlying principles for that. All right. So I've been considering this problem of measuring temperature dependence in the embryonic cell cycle regulation across sort of 3 levels of biological organization. I think by sort of breaking these down into 3 levels of organization will let me understand better sort of the minimal set of components that allows the overall embryo to scale with temperature. And so I've already shown you some of the measurements that I've made at the macroscopic level, where the cleavage divisions increase by a factor of 3.1 for every sort of 10-degree change in temperature. And I'm going to allude to the work that I've been doing sort of at the subcellular level that really allows me to exploit the properties of Xenopus laevis as being an awesome biochemical model system. All right, so I want to take a brief moment to remind you that embryonic cell cycle regulation is a lot more simple than somatic cell cycle regulation, right? There are no gap phases, there are no checkpoints. It's just a simple toggle between S phase and M phase, between replication and mitosis. And so the shortcut that I'll take is that this is driven by CDK1 activation by cyclins. And that's sort of the minimal amount of information you'll need to know about the cell cycle to sort of understand the rest of what I'm going to say. So as I said, I'm originally trained in mathematics. And so one of the things that's in my toolkit is to first build models alongside making measurements. So at the onset, I had sort of two different models for how you might go about synchronizing the embryonic cell cycle regulator. The first model I'll call constrained activation energies or constrained Q10s. And in this model, what I'm going to say is that every single reaction, every single rate limiting reaction in the cell cycle regulatory network has the same sort of temperature dependence. So I want to try to illustrate what this means. So this is a version, a schematic of the cell cycle oscillator. And what I'm going to simulate is a cell cycle oscillator where every single one of these enzymatic activities has the same temperature dependence, has the same exact Q10. And so in a simulation of this model at something like a reference temperature, say room temperature, 22 degrees Celsius, I'll get my CDK1 activity to oscillate between high and low states. So this is what a functional cell cycle regulator looks like. So now I want you to consider how this network would change once I change the environmental temperature. So if I'm cooling the environmental temperature in this model where every single reaction in that network has the same temperature dependence, it still works, right? Because they all have that same temperature dependence. I still get CDK1 activity to oscillate, albeit sort of half as fast as it did at reference temperature. Similarly, if I increase the environmental temperature, CD1K activity continues to oscillate, albeit sort of twice as fast as it does at the reference temperature. And this is again because every single one of those reactions is changing in sync. My problem sort of with this model is that it would imply a huge constraint on the evolution of proteins, right? So a priori, one doesn't expect that you would have sort of pick two enzymatic reactions and that they would have the same temperature dependence, let alone an entire network of reactions. And so I want to sort of illustrate how fragile I think that this demand is, or that this model is. So if I go back to my simulation now, and in that network, everything except one of those reactions has the exact same temperature dependence, I'm going to run simulations of this as I'm changing the environmental temperature. So again, at reference temperature, I get CDK1 activity to oscillate between high and low states, and this is good. This is a cell cycle regulatory network working. However, now when I've changed the environmental temperature and made it colder, CDK1 activity no longer oscillates. It goes into a steady state level. When I make the temperature warmer, CDK1 activity does oscillate, but quickly dampens into a steady state as well. So both of these cases are obviously not good if you're trying to construct a functional oscillatory network. And so that's sort of the problem with the constrained activation energies model, is that it really does require sort of a very strict identical Q10 sort of thing on all the enzymes in the network. So my second model is called compensatory effects from network topologies. And in this model, I'm going to say, fine, within this embryonic cell cycle regulatory system, there can be fast or there can be slow enzymes. But what pulls together the speed demons and the laggards is how the enzymes are arranged, sort of the topologies, and that there might be certain motifs, and by motifs, I mean feedback structures, that are better at worst or sort of corralling these laggards and speed demons together. And so this is work that I'm pursuing computationally. All right. So my experimental approach to try to disentangle these two models, the constrained activation energies model, or constrained Q10s, and the compensatory mechanisms approach, is to first decompose the cell cycle oscillator into a minimal course of reactions, and then to isolate and measure the temperature dependences of these in situ. So using the Xenopus-Lavis cell-free system, where the biochemical oscillations of CDK1 are intact. So I'll summarize because of time constraints, but I've done this now. I've measured all of these measurements, and I think I have an understanding, or I measured all of these enzymes, and I think I have an understanding of how the system works. I think there are definitely areas within the network in which constrained activation energies is working. This most obviously being the case where there are clearly antagonistic enzymes, such as kinases and phosphatases. But then there are other parts in the network where topological compensation from network motifs is actually happening. So even though I set up those models as being mutually exclusive, they clearly didn't have to be, and they aren't. And I think that there's a way in which to understand why we get topological compensation breaking out into this specific part of the network. And that's also work that I'm exploring experimentally. The measurements that I've made showed that there are non-identical Q10s. And this sort of leads me to another question that I've been thinking about in the background. We often think about the viability limits of organisms as being set by the properties of the proteins. So sort of at very high temperatures, proteins denaturing, and sort of at very low temperatures, proteins congealing. And so that denaturation and that congealing no longer allows life to happen. I would argue that before you get to congealing or denaturation, what actually happens first is a loss of synchrony, because no enzyme is an island, right? And so once you get certain things to happen out of sync, that happens before you get to congealing or denaturation. And so that's work that I'm actively working on now, having this sort of subset or very rich set of measurements in which to sort of toggle with. Trying to extend the viable range of Xenopus by imposing cell cycle coordination. And because Xenopus is a cool, or Lavis is a cool system in which I can toggle things biochemically, there are many strategies in which I can use to try to get the embryo to live outside of a range in which it normally does not live in. Sort of one of the things that has to happen, of course, is that you have to be able to show that the temperature-induced developmental arrests are actually reversible. And I think that I have. This is a video where I've in vitro fertilized some embryos and then move them to 4 degrees for a very long time, where the cell cycle had arrested, and then move them back to sort of room temperature and showed that they could go back to developing normally. So I've carried these embryos on into tadpole phase. So this is active work that I'm doing to sort of avert that arrest, to just get development to happen smoothly. All right, so to summarize, I've tried to tell you about how embryonic scaling and poikilothermic organisms like Xenopus laevis is sort of a non-trivial cool phenomenon. I've shown you some data of that measurement, the Q10 being about 3.1, and the Q10s for the actual cleavage divisions of the enzymatic activities, the individual enzymes, are also around that range, which is also surprising. I've shown you that what I think is happening is that there's a combination sort of constrained Q10s and topological compensation that's happening that allows the embryo to be able to scale with temperature and develop normally over this wide temperature range. And I've also argued that it's first loss of synchronization in cell cycle enzymes, as opposed to denaturation or congealing, that sets the viable temperature ranges of the embryo. And this is something I'm trying to prove actively. All right, so with that, I'd like to thank you for giving me this opportunity to share with you some of my work. I'd also like to thank my advisor, Jim Farrell, my collaborators in Leuven, my colleagues in the Farrell Lab, and also funding from the School of Medicine postdoctoral fellowship. All right, thank you.
Great talk, Connie. Thanks so much. We're a little bit behind, so unless there's a burning question, here we got one. So I'll ask you one question and then we'll move ahead. So Tony Sai asking, if you incorporate the Q10 of individual steps that you measured in your cell cycle model, does it give the Q10 of 3.1 of the cell cycle that you meschered?
This is a good question. It does actually. It does sort of depend. There is a little bit of a dependence on the model, the schematic in which you choose, because the decomposition of the measurements that we made are different. So then the different complexities of the model that you're going to simulate do matter. But yes, as a simple answer to that question.
Cool, awesome. Okay, if you have more questions, please write through the chat and Connie can, I'm sure, get to you. And without further delay, I'd like to introduce our last speaker of the 2022 Leading Edge Symposium series, Berfin Azizoğlu. Dr. Azizoğlu did her PhD at the University of Texas Southwestern in the Cleaver Lab. And currently she's a postdoctoral fellow at Stanford University in the Nussey Lab. And she's interested in understanding the principles of vascularization and then how that affects organ growth and, importantly, injury responses. So the stage is all yours, Berfin. Thank you.
Thanks, Summer, for that kind introduction. Hello, everyone. As Summer said, my name is Berfin, and I'm currently a postdoc in Rolmuza's lab at the Department of Developmental Biology at Stanford. I'm excited to share with you my research trajectory to uncover the principles of vascularization in organ size and morphology control. My passion to study this question derives from my scientific background centered around organogenesis and vascular development, and really my fascination by scaling in biology. So among animals, there is amazing variety in body size. Even within mammals with highly similar body plans, body weight varies up to 100 million times, from the famous blue whale to the Etruscan shrew at 1 pixel over here. And all these mammals you see here have more or less the same organs. So how do the organs of these animals with vastly different sizes know how big they should get? I became fascinated by this question early on and pursued to gain insight through my graduate and postdoctoral work, focusing on two main mechanisms of organ size control: autonomous and non-autonomous. So if the size of an organ is autonomously determined, the size is limited by the number of progenitors, precursor cells, within the developing organ. When precursor cell numbers are reduced, for instance, the final organ will end up smaller than normal. So during my graduate studies, I pursued to understand what autonomous mechanisms ensure proper organ size. I joined Onling Cleaver's lab at UT Southwestern, and here I focused on an organ that is thought to be under autonomous size control, the pancreas. My work demonstrated that while the size of the pancreas as a whole may be autonomously determined, this process is not autonomous at the cellular level. So using a series of genetic approaches and volumetric imaging in mouse embryos, we found that one cell compartment within the organ, the endocrine cells, depend on another cell compartment, the ducts, to reach proper mass, even though these two compartments are functionally unrelated. So when we manipulate the ducts to be stuck in an immature state, this leads to enlarged endocrine mass. So this meant that organ autonomous does not equal cell autonomous, and that the different cell types of an organ can be interdependent for size control. And these findings inspired me to go on to study organs where size is regulated non-autonomously. So here, organ size is not limited by the precursor cell numbers. When the precursor cells are reduced, for instance, the organ is regulated by extrinsic signals to make up for it, and the final organ ends up at a normal size. And this then brings up the question, how does the system know how big the organ should get? So to begin to answer this question, I joined Rolmuza's lab at Stanford for my postdoctoral studies, and I have focused on the mouse liver. And while we knew that liver size is regulated in a non-autonomous manner, we did not understand how the system knows how much the liver should grow. So to answer this, we started from the basics and asked what makes up an organ, and specifically the liver. While an organ is not merely a collection of the same cell type, unlike the earlier depictions, instead, it is a collection of functional units. So here, different cell types come together and form into a multicellular functional unit. And this unit is repeated many times to build the organ. And therefore, the principles that determine the number of functional units will also determine organ size. We wanted to know what are those principles. And to get at this, we first needed to be able to visualize the functional units of the liver, which surprisingly had not been done in mice or humans. And so here I'm showing you a thick section through the adult mouse liver, where we worked out a way to delineate the hexagon-shaped lobules units repeated throughout the organ, called lobules. And so each lobule contains a special arrangement of hepatocytes, the main liver cell type, organized around blood vessels. And this unit underlies all functions of the liver. So what determines how many of these lobules will form in a given liver? When I began this work, when and how liver lobules form was unknown. So we needed to track lobule formation. Now, the usual way to track growth in organs would be to focus on the main cell type, so hepatocytes in the liver. However, I instead focused on the vasculature, and I'll tell you why. First, like the main cell type hepatocytes, the expansion of the vasculature is absolutely necessary for the growth of the organ. Therefore, the growth patterns will be revealing. Second, tracking the vascular cells has a unique advantage because blood vessels set up the organization of the lobule, as you can see here, with the central vein in the center and portal vessels at the boundaries. So we aimed to track lobule formation by lineage tracing these blood vessels. I established and characterized a set of cream mouse lines and combined these with the MTMG reporter system. This way, we label all the cells within a vascular compartment and analyze the expansion of the compartment as a whole. So we began labeling right after birth and looked at how the cells expand by adulthood. And during this time, the liver grows by about 10 times in weight. And this analysis revealed something quite unexpected early on. When we looked at portal vessels, they rarely expanded much. On the left, you can see what the early labeling looks like, and on the right is the adult. By contrast, however, central vein cells expanded dramatically over the postnatal period. These cells gave rise to much of the vasculature within the lobule. This was very exciting. This meant that the lobule vasculature comes from cells within the vein. And moving forward, the singular source of lobule vasculature enabled us to reliably determine when and where lobules form. Imaging the whole organ, we found that not all lobules were equal. While some lobules had major expansion out of the vein, others remained mostly dormant during the postnatal time. And so in other words, the lobule on the right here has formed over the postnatal period. It is young, while the lobules on the left had completed its formation by birth, meaning it's an older lobule. We can now use this framework to pinpoint when the liver stops forming new lobules. We could lineage trace the central veins from different time points in postnatal life and compare the resulting expansion by adulthood. Once we hit a time point when no new lobules are initiated anymore, the central veins should remain dormant and not expand out, unlike the early postnatal tracing. And so here we've done exactly that. We started our central vein lineage labeling at different time points, shown here in red, and analyzed the expansion at adulthood in each case. And the top panel here shows you the major expansion from 1 day to adulthood that we've seen before. In the parallel experiment, when we induced labeling at 1 week old instead, remarkably, the expansion by adulthood is quite minimal by comparison. And so these results suggested that indeed the liver stops initiating new lobules as early as 1 week after birth. And these findings together lead us to a model for how liver size is determined. In the early postnatal liver, my work identified a population of vascular progenitors within the vein of the organ that can expand and give rise to new lobule vasculature. This process adds new units to the liver. Now, just a week later in postnatal life, the vascular progenitors do not expand anymore. No new lobules initiate, and so no more units are added to the organ. And this point of no return, no vascular progenitor expansion, and no more unit addition determines the final liver size. So then in the liver, the non-autonomous control of organ size relies on the vasculature, a body-wide cell network. So overall, the findings I've presented to you have reshaped our understanding of organ size determination. Meanwhile, they have raised a series of fundamental questions I am very excited to follow up on in my laboratory. The research in my lab will utilize this framework to understand how regenerating organs know how much to grow. And the liver provides an ideal system to study this question, as the one and only mammalian visceral organ that can regenerate. It in fact recovers its original size with high accuracy following damage, for instance, following partial removal of the organ, as you can see here. Now, while the molecular mechanisms underlying this process have been extensively studied, we do not understand how these mechanisms are integrated at the multicellular scale to eventually lead to accurate size and function recovery. And so my research program will begin to elucidate how this occurs with a focus on the vasculature. We will utilize a set of pipelines I've established during my postdoctoral work, including quantitative lineage mapping and pipelines I didn't get to share with you today, whole organ light sheet imaging and spatialomics, to study the organismal and organ scale communication principles and their underlying cellular and molecular mechanisms that ensure proper vascularization and size recovery in the uniquely regenerative liver. So with that, I'd like to thank everyone in the Nusa Lab, especially my mentor role, our collaborators, and my mentors. A special shout out to Berfin, Ian, Sherry, and Ellen, whom I've been lucky to work with, my funding sources and fellowships, especially the Leading Edge Fellows for being great community. And thank you for listening, and I'd be happy to take any questions.
Thank you, Berfin. That was a beautiful talk. So we have one question from Jen. She asks, do you think your findings are specific to the liver, or could it be broadly applied to other organs? And I assume she's talking about the growth regulation.
Yeah. I mean, as most of us are likely aware, pretty much all our organs are vascularized, and so there's a high chance that the vasculature plays into growth regulation. Now, to say whether the vasculature is the main organizer of size control, that is interesting to think about. In the liver, the vasculature is very central to the organization of the functional units, which are the lobules. That I'm not sure that would be fair to say for every organ, I know, but I can say that for certain organs like the kidney. So it's interesting to think about this. I think I would be very excited to expand this to some other organs I'm familiar with, at least pancreas, kidney.
Yeah, I have one more question. It's more, I guess, related to your future plans. In case of when you probably, I guess that's probably one of the experiments you're going to be doing, but when you remove half of the liver, let's say, the central veins are also going to be gone, I assume, right? So you think there'll be some migration or non-developmental things you observe, I assume, with the vasculature. So is that what you're thinking as well? Curious.
Yeah, and this is not known at all because the targeted lines to really lineage trace, the vessels didn't exist at all. So for all we know, the vasculature is proliferating, the endothelial cells within the vasculature. And so I think it's a really interesting question, what you're saying. When part of the central veins are already removed, are the other central veins gonna make up for it and somehow expand like crazy and make new units? Or are the units just gonna get bigger and somehow still maintain their function? And this is really one of the, I think, very exciting questions I wanna follow up on in my own lab.
Yeah, certainly. Looking forward to what you're going to come up with. All right, unless there isn't any other questions, I think we can conclude our session. I'd like to thank today's speakers and my co-host, Jen, for amazing talks and great discussion. Thank you for all the listeners. And this was the last session of the 2022 Leading Edge Symposium. So I would like to thank all the speakers and all the moderators, and specifically Kara for putting together this amazing community and all these amazing talks together. She's pulling all the heavy work behind the scenes. And I'd like to remind everyone that the talks will be on YouTube shortly through Genelia's YouTube channel. So if you missed any or want to watch again, please check out, we'll announce when they're online. And with that, thank you for listening and hope to see you next year.